All right, class, this is Mr. Nykamp, and we're going to do a quick genetics lesson today. And the lesson we're going to do today is on Punnett Squares. So I'm going to write that down real quick. And I want you to watch this video and try to learn about how Punnett Squares work. A couple things we have to do real quickly is we have to explain a couple things. First of all, in genetics, we typically represent genes with letters. And we usually choose letters that represent the trait. So for example, thinking back to Mendel in class, he looked at pea plants. And if we were talking about the height of pea plants, we would choose a letter that represents the dominant trait. So if tallness is the dominant trait in pea plants, we would use a T for tall, and we would capitalize it. So the big T represents tall, and the little t represents short. Notice that we don't use an S for short, we use a T for short, we just make it the lowercase version of it. So we would represent the alleles for tallness in a pea plant with a big T or a little t. Now also keeping in mind, parents have two genes for each trait, or two alleles for each trait. So we have three combinations of these genes we could have. We could have two dominant genes in a parent. We could have one dominant, one recessive, and then we could have a third one. My question to you is, what do you think this is? And if you said two recessive genes, that would be correct. Now, dominance and recessiveness works with the dominant gene covers up the recessive gene, if there is any. So if this was, we call this a genotype, if this was the genotype for a plant, this plant would be tall because it has at least one dominant gene, and this one actually has two. This plant would actually also be tall. This plant does not have a tall gene in it, so this is a plant that would be short. So those are some important things to know with regards to how we represent genetics with letters. Another thing, some of the terminology we use, we would call a plant, in this case, with two of the same genes, we use a term. We would call this plant homozygous. And that's a term we use for two of the same gene. We would also call this plant homozygous. And again, notice this one is two of the same genes, but they're recessive. This one is two of the same genes, but they are dominant. So we would have to identify this as either being dominant homozygous or recessive homozygous. We would have to attach that. This one, the term we use is heterozygous. And that means two different genes. And so either we refer things as homozygous or heterozygous. We should be able to determine what their genes look like and represent them with letters. Now, with that background, we're going to move on to the actual Punnett squares. A Punnett square is a tool geneticists use to determine probability. And in genetics, it's actually very simple. We draw a square, and we divide that square into 4 boxes. Based on that, sorry, let me get this in the camera here. Based on that, we have to know what we are crossing with each other, what we are crossbreeding. And so what we might say is we are crossing a heterozygous tall plant, and we're going to cross that with, let's say, a homozygous short plant. Now, we have to establish what that means as far as the genes go. If you look back at the notes I gave previously, we would represent a heterozygous tall plant with a big T and little t. Heterozygous, again, means one of each gene. So this would be describing one of the parents in the cross. And homozygous short would be little t, little t. This is the only way we can describe those. We would then take these parents and we would put them into our Punnett square grid. And the way we do this is we would take, let me redraw this. If we have one parent, big T, little t, this is the heterozygous parent, crossed with a homozygous small or short plant, we would take one parent and we would split their genes up, because this is what meiosis does. We learned this earlier. Meiosis splits the genes up. And we would write, and it doesn't matter, for this example, I'm going to write it right on the side here. We would write the genes down the side of the box. So I wrote a big T, little t, that came from one of the parents. I would then take this parent here, and I would write them across the top of the Punnett square. So I would do little t, little t. From there, I would look at the combination possibilities. We do that by kind of following our fingers across here. I could get a little t from this parent and a big T from this parent. So this offspring would have a heterozygous genotype. If I go across here, I would get a big T and little t, following that T across and that T across. This is also heterozygous. If I go down here, I would get little t from this parent and little t from this parent. And I would get little t and little t from this parent. Now this only works with traits that show dominant and recessiveness. And so what this does is it gives us a probability of what we would see in the offspring. If you look here and you start to understand what the genes mean, these two would be tall plants because they have the dominant tall gene. So we would have a 2, let's see here, where can I write this? A 2 out of 4 chance to be tall. 2 out of 4 chance to be tall. If you know your percentages, that would be 50% chance to be tall. And that is what the Punnett square tells us. Here we have a 50% chance to also be short. And so if we crossed, going back to our original cross here, a heterozygous tall plant with a short plant, we would have a 50-50 chance of getting a tall or short plant. So if you planted a bunch of seeds from that cross, you would get 50% plants that are tall and 50% plants that are short. We have to know the genetic makeup of the parents to determine that. Once we do, we can plug it into a Punnett square, very much like we did here, and try to figure that out. So Punnett squares are tools used to determine probability in genetics. We are going to go over this in class tomorrow as well. So hopefully watching this video will get you started with the idea, and we'll talk about this tomorrow.